I just want to start my talk with this slide. It was actually in 1959, Richard Feynman, who is one of the Nobel laureates, actually gave a speech at Caltech called "There is Plenty of Room at the Bottom." This actually officially started the advance of nanotechnology. And I'm very grateful for him, for his work, not only for nanotechnology initiation, but also the other things that he has done in actually physics. So today, what I'm going to talk to you is how actually we can improve current radiation therapy using nanotechnology. That's in a short way, that's how I would like to explain. And so what is nano? I'm sure you must know, but just to get everybody on board, if you think about it this way, if you take a drop of blood, it has actually 5 million red blood cells, 5 million. And if you look at a red blood cell, and the diameter is 2,500 nanometer. And if you look at the DNA, the width of the DNA complementary strands, it's basically 2 nanometers. So now you can imagine how small is this nano scale is. So now the question is, how can we use this nanotechnology to cancer therapy in terms of before we go into radiation therapy? Why do we think that we can actually exploit nanotechnology, or in other words, nanoparticles into current medicine? So this is actually a cross-section of a cell. We are made out of trillions of these cells. So this is basically the nucleus, and this is the membrane of a cell. And this is like a small factory, if you want to put it that way. So you have a lot of organelles inside doing different things. And if these cells was to function, that's why we take food. So what do you mean by food? That means we take proteins and other things for us to, survive. So these protein molecules actually can get into these cells, right? At the same time, we get sick when the viruses attack us. So the viruses are basically 70 to 100 nanometer in size. So I'm sure you're familiar with the coronavirus, which is about 70 nanometer. So that's how we know that if we can make something in 10 to 100 nanometer, we can actually send them inside cells to do something useful in terms of cancer therapy. So how far we have gone in that direction? So people actually use nanoparticles to deliver drugs and also imaging contrast agents and so forth. So we have built actually library of these nanostructures, lipid-based, polymer-based, and of course drug conjugates, viral nanoparticles. But my focus today will be inorganic nanoparticles, and especially I'm going to talk to you about gold nanoparticles for a very good reason. When I talk about gold, what do you think? Do you think it's yellow, or you think it's this red? So usually when we talk about gold, gold is yellow, but when we make nanoparticles, it turns into this color. So just to give you an idea. And then the reason I chose gold nanoparticles are for 2 reasons. One is gold nanoparticles can be made with different sizes. 10 to 100 nanometer, you can make it, right? And then you can also functionalize them easily. And and it is biocompatible. And it is actually used in phase 1 clinical trial. So that means we can actually have the probability to use it in humans too. So now the question comes is if we were to use these nanoparticles in cancer research or any other medical application, will the size of these nanoparticles really matters? Or in another way, if I ask the question, can I just use a 10 nanometer gold nanoparticle or maybe a 50 nanometer gold? Does it really matter? That was one of the questions, right? So the gold turned out to be a good materials to understand that, because by changing the gold source and the reducing agent, the ratio, you can make different size nanoparticles. So you use the same material system if you want to understand the effect of size. So what do you think you would see? So before I give you the output, I want to explain something very simple. When I talk about these nanoparticles and talk about how the size affect, you want to know how these nanoparticles can enter cells and go through cells. So what we believe is that this is a gold nanoparticle. For example, these red circles are all gold. So we believe they actually enter cells through endocytosis process, trap in these organelles, and then of course at the end excreted through the cells. So this is one of the major mechanisms of gold nanoparticles entering the cells and how they travel through the cell. So now, how do you visualize it? This is a schematic. So you will see throughout my talk different images to show nanoparticles in cell. So this is actually a TEM image, transmission electron microscopy image. This is actually the nucleus of the cells. This is what you have, the nucleus of the cell, the brain of the cell. I told you these nanoparticles get trapped in these organelles. So these are gold. So in a TEM image, if you look at each one of these organelles here, are here, and these black dots are basically gold. This is a transmission electron microscopy image. And this is a confocal image. If you were to look at these nanoparticles, these vesicles actually appear as red dots here throughout the cell. And these red ones are gold nanoparticle clusters. What is this in green? Is the actually microtubules in the cells. These are basically highways in the cells that actually traffic these vesicles in the cells. That's a very interesting thing to learn. I will come back to that later. So this is a confocal image where you need to label gold nanoparticle with the optical probe. We also use another technique which is called dark field imaging, where you don't have to label gold nanoparticle with the optical probe. So these bright structures you see are all these vesicles containing gold. And how do you know these ones are gold? So basically it has a function where if you go to these bright dots, you can see the reflectance spectrum of gold. Of course, if you go to the background, you can see it is flat. So these are the three techniques we use actually to see gold nanoparticle in the cells. Now we know how to see them. Now the question we need to answer, how the size of these gold nanoparticles affect how they internalize in these cells. So what we did was we actually look at it at a monolayer level, 2D. So what we noticed was the gold nanoparticle actually, if you look at it, the size has an effect how they get into the cells. For example, if I use here 10 to 100 nanometer gold, you can see 50 nanometer gold has the optimum internalization in the cells. And then these are of course TM images capturing different process that I discussed before, right? So this is the endocytosis process is captured here in the TM images. This is the gold is here. So you can see these organelles with gold nanoparticles here. And this is of course this excretion process is given here. So this is all good. And then, so that means there is an optimum size. Now, after I published this paper, there were so many theoretical papers came out. Actually, they showed that this is really true theoretically. In another way, if you wonder why there is an effect of size, it is actually due to the competition between receptor diffusion on the membrane of the cells, as well as the energy that it's necessary for nanoparticles to for the cells to actually wrap these nanoparticles for them to enter. So when the nanoparticle is very small, it takes so much energy for the membrane to wrap it. So it is not favorable. So they want to be like a clustered on the membrane, right? To reach that point. So that is a probabilistic process. When the nanoparticles are really large, it consumes a lot of receptors. So that actually end up with less receptors for the other incoming nanoparticles. So that's why there is a optimum size in the middle. So this is actually all good. Now the question is, this is a fundamental result. Now you want to see how am I going to capitalize this fundamental result into an application. So that's what you want to do next. So I actually joined Princess Margaret Cancer Center to test this fundamental study into an application. So there, I actually, what I did was I used gold nanoparticles as a radiosensitizer. How? That is, the gold nanoparticle actually is a high atomic number material. High atomic number is 79, which is high, right? And it actually can interact with radiation. Radiation means photons. So these are basically photons. So the gold nanoparticles can interact with photons and actually send us a shower of secondary electrons. So these secondary electrons actually, what do? These secondary electrons actually can interact water molecules. 90% of our cells are actually water, right? If you look at it. So as a result of that interaction, it produces more radicals. So the red ones are extra free radicals produced in the presence of the gold nanoparticle, which actually can lead to higher radiosensitization in the presence of a radiation beam. So this is the rationale behind using gold nanoparticles as a radiosensitizer in cancer therapy. So now, is this the only mechanism? No, there are other things that we really don't know yet, but we are looking into these aspects. So I'm going to give you this picture. This is a schematic in some ways, but I'm going to give you a different explanation so you can understand it better. So now, just to, if you are not familiar, this is actually just to show you, this is a tumor, basically. Consider this is a tumor lesion now, for example. So what you want to do, you want to treat this tumor lesion with radiation therapy. So that means you have a photon beam, light beam, right? Basically with the high energy. So the X-rays means these are X-rays with high energy. That's what we use to treat patients. So if this is the tumor lesion, what we are trying to do, we are going to send these photons from different directions in a way so that you try to exposure. This is the normal cells surrounding normal tissue. Try to reduce the exposure to normal cells, but at the same time, every time you are targeting the tumor. So that's why if you look at a linear accelerator, this is the patient, you can see it can rotate. So the idea here is to reduce the normal tissue toxicity while targeting the tumor. So what is the theory behind now? So I want you to focus on this graph. So there are 3 curves here. So I want you to focus on these 2. So the dotted curve is actually normal tissue. That's very important, right? Surrounding normal tissue complication probability. And the middle curve actually is the tumor control probability. Now think about it. You want to have 80% control of the tumor. Now, when you try to do that, see what happens. You get the control 80% with the tumor, but at the same time, you still have a pretty high normal tissue complication probability. So what you want to do is to reduce this normal tissue complication to a lower, like somewhere here. How do you do that? So in order to do that, that's what we do is that we can actually put a radiosensitizer in this tumor, right? So when you add a radiosensitizer, for example, gold nanoparticle, what it does is that it's actually it can push this tumor control probability to the left, because it can actually enhance the dose, the effective dose. So you can basically, what you are doing is that you can reduce the normal applied dose to a certain extent. So you can see in that case, normal tissue complication is much less. So our goal is actually increase the dose to the tumor, at the same time reduce the toxicity to the normal tissue. That's one of the side effects of radiation therapy. So that's basically our goal. Now, what I want to ask you is this done? Is this tested? Yes, it has been tested. That means in a mouse model, gold nanoparticles as a radiation sensitizer has been tested. If you look at this graph, you really can see here. So this is in a mouse model. you can see if you use gold nanoparticle with radiation, you can see actually they were able to bring the tumor volume almost to the 0. And this is only radiation alone. This is very effective. Then the question is, it was done in 2004. Why it's not in the clinic if this is that good? So what is the issue here? The issue here is two things. One is the concentration of gold used is the 5 milligram of gold per gram of mouse. So which is actually pretty high. We cannot actually do that clinically. It's impossible to do. You need so much gold, and it's not practical. And also the energy that used for this radiation is actually in low energy, KeV. But you cannot use low energy in a patient if the tumor is deep-seated. That means you have to use higher energy. So then how are you going to achieve those two objects? That's where I'm trying to do. So the question is, why? How can you explain this result with what I have learned already from my fundamental study? Why do you think they needed a very high concentration of gold? That's one of the questions I was trying to figure out. So that means they use 1.9 nanometer gold, which is very, very small. I think if I talked to you before, gold nanoparticle going into the cells is dependent on its size. So that when the nanoparticles is really small, the uptake is very, very low. So that means that's why you have to use a very high concentration to arrive at the concentration to do sensitization. So that means if I can actually optimize the size of the gold, we should be able to reduce the amount of gold that we need for sensitization. That is what I did actually. So by optimizing the size, you can actually reduce the gold by a factor of 1,000. So you can see I was able to reduce the concentration necessary by a factor of 1,000. Now I'm going to show you some data. So this is the statement I made. That here, so I use 3 different sizes: 14 nanometer, 50 nanometer, and 74 nanometer. So this is how I did the experiment. So this is actually a low energy KeV unit. It's called, in clinic we call it auto voltage unit. And then this is where the sample with the cells, and so these cells have gold nanoparticles or no gold nanoparticles depending on the condition. And then we treat them with the radiation dose, and then we do a clonogenic assay to figure out the survival fraction. So in this graph, what you have is the y-axis is the survival fraction, and the x-axis is basically the dose. So I want you to focus on 8 gray here. For simplicity, we do different doses, 8 gray of radiation dose. And then what are these curves? So the black curve is basically the control cells. That means the cells we gave a 8 gray dose, but it does not have gold. This one, the red one, is actually the cells with gold nanoparticles of size 50 nanometer, 50 nanometer diameter, which has the optimum uptake, right? And these two represent cells which are smaller and larger nanoparticles. You can see having more gold nanoparticles in the cells, as we expected, will actually enhance the radiation sensitization. So this is 50 nanometer. You can see highest sensitization and the lowest survival fraction, which is pretty good. And I did it at a nanomolar concentration, and which is very low. So that is one of the things I want to get through to you. You don't have to do experiment at concentrations pretty high. What you want to do at concentration in your labs, in a concentration that you think you can achieve in vivo, right? So then you don't have to go to the drawing table back and forth. The concentration I work right now can be achieved in vivo. So that means I know this can be translated, you know, at some point, and it makes our life easier. So by training, I am a physicist. When I see this biological data, I really want to see, is there another way to test the same thing, you know? And then what I did was that I actually look at what is happening at DNA level. Remember, I talked to you about sensitization of cells due to gold nanoparticles, how the DNA can be affected. So here we did the experiment, and actually we probed the DNA double strand breaks. Why double strand breaks? because it's the most lethal one. So you can see these circles here. These are the nuclei of cells. So this is a nucleus. This is a nucleus likewise. So the top row you see is actually the cells with no gold nanoparticles. The bottom one is the cells with gold nanoparticles. So you can see, so we actually look at two DNA repair proteins that actually present in a DNA damage. So gamma H2X and 53BP1 to be sure, right? So we prepare two, and then we actually merge the foci here. And then what you can see right away, in the presence of the gold nanoparticle, it actually brings more foci, damage sites compared to cells with no gold. So this is actually in agreement with the cell survival that we saw before. So this is very good. When we actually did this one not with the lower energy. So you will ask the question, yeah, you complain about lower energy. What about the energy that we use in the clinic, which is the linear accelerator? Will it work? That is the biggest question here, right? So this is the linear accelerator. So this is where the sample is. You put solid water to do the treatment plan here, like a patient. And then what you see here is the top row is cells with no gold. Bottom row is cells with gold with two different repair proteins probed. So you can see after 24 hours of the treatment, this is permanent damage. You can see cells with gold has more foci compared to cells with no gold, which is significant. And when we published this work, actually, it was featured on the cover of Radiation Research. That really shows how significant this work is. And it was also awarded the Michael Patterson Award by Canadian Organization of Medical Physicists, considering the impact it made in the field, which is very good. Now, I want to summarize what I have said so far so that you can breathe a little bit. So what I said first was yes, the size of the nanoparticles matters. And I said there is an optimum size. So that's good. And I also said gold nanoparticles can be used as a radiosensitizing agent. Yes. So this is the cells with no gold. Cells with gold, you can see higher number of damage, nuclear damage in the cells treated with gold. So that is telling, when you look at this image. Now, there is a question now. However, all of this work done at a monolayer level. That means 2D, two-dimensional. So you can see these cells, and but the question is, what happens at the tissue level or in animal models? Can we answer that question? Will you see the same effects in tissue levels and the 3D level? So that's very important, right? So before I go into animal models, I want to talk to you about three-dimensional tissue models. Why do I focus on three-dimensional tissue models? That's a question. So here I talked to you before. So far I talked to you about two-dimensional models, that is monolayer of cells. You can see there is no barrier between cells, so that any therapeutic agent is accessible to these cells, right? But what happens when you inject these gold nanoparticles, for example, for a patient or an animal through the tail vein? So they have to go through the blood vessels and go through these blood vessels and in the tumor blood vessels. So what happened in a tumor blood vessel? In a tumor blood vessels, we know that tumor blood vessels have some sort of leakiness. So we expect these gold nanoparticles to leak out of these tumor cells and enter the tumor tissue. So the question is, how do these nanoparticles go through this tumor tissue, right? Is it like these ones, like a monolayer? Is it so simple? That's a question to answer. So here, what you see is, this is a tissue cross-section, right? So the marked in grease is actually the extracellular matrix. So we did the same treatment here, but you don't see this barrier in the monolayer of cells, but you see how much barrier is there in tissue. So that means you have to see whether nanoparticles can overcome this barrier. So these yellow bright dots are basically all gold in the tissue matrix. Now, the question is, how do you understand this thing in a 3D tissue models? How do you make 3D tissue models? Is there one way or is there another way? So what I did was I used two different types to see whether I can see the same effect at the end of the day. So this is one model I use, that is, we can actually get this thing. It's called molecular inserts. It's available. It has a membrane at the bottom with holes so that it can actually get the food like media. So what you do, you put a monolayer of cells at here, and then you can grow these one into a tissue layer. Like for example, here is a grown tissue like that. You can see from this is the bottom, this is the top, it is a tissue layer, tissue cross section. You can see all the dots here, bright dots are all gold from dark field imaging, remember I talked to you about. So this is actually marked in green is the extracellular matrix that you see. And so this is one way I did. I published this work, and it was actually featured on the cover of Nano Micro Letters, and which is good. And then we came up with another model that is, a lot of people are familiar with the tumor spheroid model. So if this is going to I just wanted to see the outcome I gained from this model. Is it similar to what if I have another 3D model? Will it be the same? To be to tell you the jump the gun here, yes, we saw the same thing, which was very good. And so in this case, we make a tumor spheroid, and this is just to show an image of a tumor spheroid. And then we actually can, if you look at that in a tumor spheroid, you can see the gold nanoparticles are in the spheroid. This is an unstained image, dark field image, something similar to here. And this is a eosine stain section of the spheroid. So you can see here, similar to here, you can see the extracellular matrix and stuff. And so these are two models we consider. And the question is now, what is the outcome, right? What is the outcome? So in the monolayer level, you saw there was an effect in the size. So you can see that, you remember, I think you already know that. Now what happened at the tissue level? So what we note was in the tissue level, of course, you see the extracellular matrix. We saw something very interesting. That is, at the monolayer level, if I use 20 nanometer and 50 nanometer, as you expect, 50 nanometer has the highest uptake compared to 20 nanometer. So that's what we see here. We use two different cell lines, two breast cancer cell lines. And then what happened at the tissue level? So you can see at the tissue level, actually, smaller nanoparticles are beneficial compared to 50 nanometer particles. That is interesting. So now, how do you do it? Now I'll explain it to you. But we know 50 nanometer is the highest uptake compared to smaller. But when you go into tissue, you can see these smaller nanoparticles are much better actually. This is this is actually something to do with the penetration. If you look at it, small nanoparticles can penetrate better with the extracellular matrix. That's why. So just to summarize, what happened at the tissue is this diagram actually, which is in the cover of the journal. This was featured 2 times actually, this work. So this was one of the covers. So what we have here is that at monolayer level, smaller nanoparticles are localized less compared to 50 nanometer gold. But if you look at the tissue level, smaller nanoparticles penetrate better compared to larger nanoparticles. Now, how do you solve this puzzle? So which one is better? You want to use 50 nanometer or smaller nanoparticles? So that was, we had to go back to the drawing table. And then what we did was we did something interesting. That is, we took the small gold nanoparticle and of course 50 nanometer gold. We actually labeled them with a natural, it's a peptide which has an integrin binding domain. Why is that? RGD is an integrin binding domain which actually expressed in most of the tumor cells. So it is basically what you are trying to do is the targeting, but you cannot put RGD peptide onto the gold without a stabilizer. So considering that we want to do in vivo studies and humans later on, we thought the best way to use is that adding a polyethylene glycol molecule. Something interesting happens after this. So what happened was so we did this thing for 50 nanometer gold, smaller, and 50 nanometer gold, 50. What we noticed was we were able to increase the small nanoparticle uptake by a quite amount compared to 50. Interesting, right? So now we know 50 nanometer particle, when they reach single cells, they can internalize better, but at the same time, they can actually penetrate through tumor tissue also better. So which is a good outcome. So then can you explain this? That's the question. Can you really explain it? The way we explain it is this. If it is 15 small nanoparticles was 50, what we think is happening is the small nanoparticles have a curvature is high compared to 50. So what we think happening is that this the RGD peptide can access the surface receptors on the cells better compared to 50 nanometer, where it is kind of screened because the RGD peptide is slightly smaller than the PEG molecules. So we think that was what's happening. And then this is all published, you can see in these papers. And then we went into 3D tissue model now. This is at monolayer level, 2D. When we tested in the tissues, what we noticed was that, of course, 15 nanometer particle has a better penetration and the uptake now compared to 50 nanometer. So now that is interesting now. So we know it works at 2D. We know it works at 3D. So the next thing you want to test is, will this really work in an animal model? So what we did, that's exactly what we did next. So we actually had an animal model, and we injected our gold through the tail vein. And then, of course, you have a subcutaneously grown tumor. And then, so we expect these nanoparticles to go through the blood vessels and enter tumor blood vessels and enter tumor tissue. And then, as a function of time, if you look at it, accumulation of gold nanoparticles in the tumor, what you see is that at the 24 time point, you have the maximum amount of gold within the tumor tissue, which is a good thing. So we have enough time in an experiment to do radiation therapy and so forth, right? You can get ready to do the treatment process. This is looking into the biodistribution first, right? We want to know at what time point you want to give the radiation dose. That is when you have the maximum amount of gold. That's what we are trying to find out in this biodistribution study. So do you have any proof whether gold is in the tumor? We do actually. So this is actually just to show a tumor blood vessel. So you can see that it's like a cut cross. You can see and then you can see the yellowish structures are all gold. So these are basically blood vessels cut the other way. So you can see just the top, like cut across, and these are blood vessels here. And of course, these are cells. You can see closer to the blood vessel, you can see a lot of gold in the cells. So that is very encouraging. Now I'm going to close this story here, where I just wanted to show you how important it is to test something in 2D and the tissue level before we testing in vivo. It works. But now the question now I'm trying to address is when we have this kind of a study where we have a solid tumor here, we use standard cell lines. But we are missing some of the factors here. You don't see some of the things that you see in a patient tumor. What are those? So I'm going to get into that. I'm going to go a little bit deeper now. So that means when you think about a tumor, are you considering only the tumor cells or cancer cells, you call it, right? Are we have to worry about only tumor cells? No, there are other things we need to worry about, which is tumor microenvironment, right? So our most two interesting factors are tumor tumor normal fibroblasts as well as cancer associated fibroblasts. Why these two are significant? So based on some of the information we have is that these fibroblasts exist, it's everywhere in our tissue, right? So the closer to the tumor cells, actually, at the beginning, you have these fibroblasts too. So what happened in this process of tumor growing is that some of these fibroblasts are recruited by tumor cells and turn them into something called cancer-associated fibroblasts. But these two are behave very differently. So these actually cancer-associated fibroblasts, I call it CAF, 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 cancer-associated fibroblasts, actually promote tumor growth. So my question is here is that if you try to find a therapeutic approach to kill cancer cells or tumor cells, that means you have to know how it's going to affect these cancer-associated fibroblasts too, right? Because it promotes tumor growth. That's exactly what we were trying to find out. We have access to patient-derived these CAF cell lines too. So we did a very interesting experiment. So where we actually look at how these gold nanoparticles go into fibroblast, which is I have here normal fibroblast, HeLa, which is a tumor cell line here, and this is CAF, cancer-associated fiber. Interestingly, you have more gold in the cancer-associated fibroblast, which is, I believe, which is a good outcome. So what we want to do is that can we use the gold in this cancer-associated fibroblast cells now to reduce its activity so that we can actually benefit when we do radiate these tumor cells, we can kill these cancer cells. So that's the argument here. So do you see what you see here in images as well? This is cancer-associated fibroblast cells. You can say they are bigger and they have more gold, like you see here, right? And this is cancer cells. All the bright structures are gold. So this is tumor cells. This is normal fibroblast. It has lower uptake. And now I want to show you what is the outcome now when you do radiation. So these actually confocal images. You can see cancer-associated fibroblasts. You can see more gold in them. The green is the microtubules. This is normal cells, fibroblasts. This is tumor cells. And all red structures are gold. And then what we did here, this is the uptake I told you. So it actually translates into a therapeutic response where we see more DNA double-strand breaks with radiation in the cancer-associated fibroblasts and tumor cells versus normal fibroblasts, right? So and we did proliferation studies. What we noticed was the proliferation, the HeLa proliferation and the CAF proliferation, they were all affected by radiation therapy and having gold in them. Now, what I want to tell you one more story is that, but we did these experiments using separate cells, right? CAF separately, HeLa, the tumor cells separately, fibroblast cells separately, but that's not the right way to do it. If you were to do it right, what you have to do is to see the crosstalk between CAF and the tumor cells, you have to do them in co-culture, right? To see the effect of CAF on the therapeutic outcome of cancer cells, which is exactly what we are doing, right? And we noticed that having such number of gold in the CAF, we were able to actually using that to reduce the activity of CAF that actually, and we were able to promote the outcome of radiation in tumor cells, which is actually very nice study. I'm going to talk about that in detail in the pancreatic research round next month. And if you want to listen to that detail, more pancreatic cancer-oriented research, you can actually reach out to me. I will put you in contact with Pancreas Center BC. But I thought this is a general talk. I'm not going to go into so much into details. So we are doing a lot of work with co-culture and separating them. It's a very fun study. So now we talked to you about cancer cells alone in 2D, 3D, and animal models. And we talk about that, okay, tumor is not just the tumor cells, it has a tumor microenvironment. How these gold nanoparticles are interacting with tumor microenvironment. Can you control that microenvironment as well? So we see the positive signs of it. Now, I'm going to shift gears a little bit to show what else we can do. So far I talked to you about how I can use gold nanoparticle with radiation therapy, for example, right? The outcome. So the next thing I want to talk to you is, can we be a little bit smarter than that? Can I use some other things? So here what I'm going to show you here is so this is the tumor lesion. So I can put gold in the tumor lesion, for example, think about it that way. What else I can put it in this tumor lesion so that I can benefit more in the presence of radiation? So that's where some of the chemotherapeutic drugs come in. So they are clinically available. So one of the drug is the docetaxel that we use in metastatic cancers. So docetaxel is a very interesting and very important agent for our work. And I'm going to explain it to you how I can combine this docetaxel drug with gold nanoparticles to exploit the full potential of radiation therapy now. I'm going to track the time. So here, what is happening here is what I'm trying to explain it to you here is when you have the docetaxel, it does something very unusual, very important. It actually blocks the cells in something called, this is the control cell population. It blocks the cells in a more sensitive phase to radiation, G2M phase. And also, in the presence of the docetaxel, what it does is actually the microtubules in the cells are bundled, like you can see here, bundles. So docetaxel can actually stabilize these highways in the cells. So in a way that push gold nanoparticles towards the nucleus of the cancer cell. So this is very interesting now, right? When you do radiate, so you can get a shower of secondary lectin to the nucleus to do the DNA damage. That's exactly what you want. And then if you can tell me you show only one cell. No, this is actually normal cells. The nanoparticles distributed throughout. With the docetaxel, you can bring them closer to the nucleus. These are two different images showing how we can concentrate gold. And there are a lot of other benefits to having gold nanoparticle in cancer cell using with gold. Is that one is the uptake is gold is going to be increased. I think it is more throughout retention. So gold nanoparticles are retained better in cells with the docetaxel, which is very, very critical for us to do basically fractionated treatment in the clinic one day. So the question now here, so how does it work with radiation? Now we are going to have the two gold nanoparticles and docetaxel. So what you see here is that with gold nanoparticles and docetaxel, you can see synergistic effect, which is pretty good actually. This is exactly what we wanted. But I want you to remember this is at 2D level. Still, this is now I've proven it monolayer level. So the question here is that what happened at tissue level and in vivo level to this kind of a therapeutic option? Those are the questions. Not all questions I can answer, but I can answer some of the questions. So how does gold nanoparticles behave with docetaxel in a three-dimensional environment? So we grow tumor spheroids, and what we noticed was something very interesting. That is, with the presence of the gold, you can have more. With the presence of the docetaxel, you get more gold nanoparticle trapped in the tumor spheroids, two different types of cell lines, which is what we saw in 2D as well. We see the same thing in 3D. So the question is, what happened in an animal model? That's the most beautiful part. So in an animal model, we recently, this is under review with the Nature Scientific Reports. So what we noticed was that with the presence of the gold nanoparticles, we can actually increase the presence of the gold nanoparticle within the tumor over 100%, which is pretty significant number. And so this is the control with no docetaxel. This is gold nanoparticle with docetaxel. You can see all the bright dots are gold, which is actually you really can see. Now, actually, to be honest, to give you the good news is that we are as we speak, we are doing the radiation study at the Mayo Clinic in US because we are still working here to get the approvals. So now this is actually to show the cell cycle in the tumor. You can see it is actually even within the tumor in vivo, cells get arrested in the most sensitive phase to radiation, G2M, which is very interesting. So we are doing in vivo studies right now. What are the other platforms that I'm interested in? Is we actually did one platform with Peter Cullis where we put gold nanoparticles in a lipid system. We were able to increase the uptake of gold in the cells, which is good. And now we actually did the drug, one of the drugs in the lipid system. We were able to deliver them very nicely, even in vivo, which I talked to you about in the pancreas research rounds. Now our final goal is to put both the drug and the gold in a lipid system and to do this system. So two messages to carry is one is that the combination of gold nanoparticles and very unique combination of gold nanoparticles and chemotherapeutic drug. For example, docetaxel can be used in a smart way to actually improve current radiotherapy. And of course, it is better if you can test your therapeutic options in 2D, 3D level before you testing in the animal model. So I work with a lot of collaborators in US and across the world. And so we wrote two roadmaps to bring this to the clinic. And it is heartbreaking to show this one. This is my mentor, Dr. Richard P. Hill, who passed away recently. I was one of the very lucky ones to have someone like him as a mentor. Like a, you know, I miss him so much. So, so here, this is my group. A lot of young scholars. I treat them like all my kids, and so they are doing really well, helping me. And these are the branding agencies who help me unbelievably. And so these are very good group of people. A lot of them belong to a nanomedicine network, especially Nancy. I do a lot of animal work. And just to mention the Dr. Alexander Abraham, I never design experiments without talking to him. He's a radiation oncologist. This is a radiation oncologist, medical oncologist, and of course our two medical physicists, and all of these people. And this is my young star. I took this when I went to Disney World. She wanted to be a clinician scientist one day. I said, okay, but I didn't show my enthusiasm to see whether she would change her mind. Now she is in medical school. And to be honest with you, I'm thrilled. So I hope she's interested in oncology on top of that. And then I'm looking forward to I put this one to make everybody jealous, because this is where I live. Thank you.
Oh, that's fantastic, Devika. What a wonderful talk. And thank you for sharing so much about your life here at the end as well. Congratulations to you, a proud mom of your daughter. I remember meeting her, and she's obviously done so well. That's just wonderful to hear. And so the science that you presented today is just fantastic. And I think for anyone that's not working on gold nanoparticles, this also makes it very accessible for them to really understand why you're interested in this space, which is great. So we'll move now to taking any questions that there may be. And we do have one by Liza Silverman, and Liza's asking, is there an issue with bioaccumulation with gold nanoparticles? So I'm not sure if this is related to potential toxicity or actually achieving that accumulation at that target site, but perhaps you could comment on both.
Yeah, that's a very good question. Actually, I'm also looking at the clinical feasibility too, right? When I work on mouse models, I want to see the in vivo accumulation of gold nanoparticle in tumor tissues is good enough to show sensitization even in humans, right? So we are using a concentration that can be used in humans. It's because we actually use 2 milligrams per gram, 2 micrograms per gram of mouse. So you can imagine. So we did a calculation today how much it would cost me to do a human. So it's like less than $10. And the kind of accumulation you have is not toxic based on animal models. But there is a phase 1 clinical trial already done, right? So we are within those limits, everything. So we believe this can be translated easily, and that's the goal actually with our radiation oncologists, 2 radiation oncologists, one from US, one from Canada. So we are planning to do one very soon. At some point, you know, I'm waiting.
Wonderful. Thank you, Devika. Any other questions? I recognize lots of names here on the attendee list, which is great to see. Folks, I can see that you worked with a long time ago and now currently, so that's nice to see. Maybe, you know, one of the things that I always have been challenged with when we did the gold nanoparticle, we'd done some gold nanoparticle work, was intravenous administration and achieving sufficient tumor accumulation following intravenous administration. And in fact, in a number of cases, we just ended up doing intratumoral injections, which I don't love to do, just in terms of clinical feasibility moving forward. Can you comment on that? Like, do you think there are challenges there moving forward?
I think it depends how you functionalize your nanoparticles too. And but I don't believe in targeting nanoparticles to the at the beginning. Once it reaches the tumor, only we worry about the targeting, because targeting happens once it reaches the tumor, right? So it depends how you functionalize these nanoparticles. And one of the things, that's why I brought this docetaxel issue here. So that is one of the drugs we can actually use to trap the gold nanoparticle within the tumor. We have been able to trap it over 72 hours. So we didn't go beyond that, but we believe it has a very good retention too. So that means you don't have to do many injections, right? So docetaxel is given weekly to humans, right? So for the same reason.
Great, thank you. And I can see that I do have more questions coming up. Oh, here's my, it's because I have another screen open here, sorry. So we have another question from Jennifer Zapata. Do you have important accumulation of nanoparticles in other tissues and organs? Do you have?
Yes, very good. Actually, the paper is coming out. It has in liver, spleen, and all of these. But I try to stress this point. We are injecting a very small amount of gold, but it will result in enough accumulation in the tumor for sensitization. But you have to remember, we are doing a local radiation, right? So even if it is in these organs, it should not affect you actually. So that is, and we don't see any toxicity in those organs with, we will do a very long-term study. Short-term, we don't see any toxicity. And yeah, good question.
Okay, good, thank you. And then maybe just one last comment and question here. So this person's saying incredible talk, and they're wondering if the equipment used for radiotherapy in humans would be enough to use in deep tissues with cancer.
Yeah. So actually, the linear accelerator I used was actually it is used to treat patients. So I never do with the small animal, the radiator. I would say, because the interactions of gold is very different from low energy versus MEV, right? So we usually use actually the cancer machines at the BC Cancer. I have one, the head of the medical physics at the Victoria, and a head of the medical physics in Vancouver, doing in vivo work. In vitro here in Victoria, we never use other machines. We all use clinical machines. Thank you. That's a good question.
Great, thank you. Well, I have to say, I really enjoyed this talk today. It's great to see just the full picture of everything that you've been pursuing over the last several years. I've heard from many people that you are an amazing lecturer, and I can see that in the way that you presented today. I mean, everything is just so clear, and you can see your passion for science coming through, and that's always been the case. So really inspiring to hear your talk today, Devika. Thank you so very much.